Now, the neuroprotective effect of tualang honey on kainic acid-induced neurodegeneration in rat lateral receptor region. So briefly, kainic acid aspirin model has been widely used to study the mechanism behind various types of neurodegenerative disease. Upon binding kainic acid on glutamic receptors, it will trigger serious cascade of events which eventually result in neural cell death. And tualang honey is rich in phenolic compounds and has a potential further effect on different brain regions. However, the effects of tonal honey on kainic acid-induced neurodegeneration in retroceptor brain region hasn't been studied. So based on in the study, the rats will be randomly divided into four groups, and each will be treated for 5 times at 12-hour intervals, 30 minutes after the last oral treatment. The rats were injected with kainic acid and observed for seizure characteristics. The rats will be sacrificed at 24 hours or 5 days after kainic acid administration. So based on the result, first this H&E staining shows control group with normal rounded to oval shaped neuronal cells indicated by the black arrow. Meanwhile, in kainic acid group, features of generative neurons were observed, and pretreatment with tolahani or topiramate ameliorated kainic acid-induced morphology changes when neurons appear comparable to control. And then the neurons were further quantified. Based on the crystal bodies analysis, the number of viable neurons in kainic acid group were significantly reduced when compared to control at both time points. Moreover, the number of neurodegenerative cells in kainic acid group based on FJC staining were significantly higher when compared to control. Meanwhile, in pretreated groups, the number of neurodegenerative cells was significantly reduced when compared to kainic acid in 24-hour subgroup, and similar trend was observed in 5 days. So in conclusion, this result shows that kainic acid-induced neurodegeneration in receptor region and tualang honey might have potential in protecting lead receptor against kainic acid-induced neurodegeneration. However, further study need to be done. And thank you. That's all from me. And this study was financially supported by Universiti Sains Malaysia. Thank you.